Hello everyone, this is Navdha Soni, Assistant Professor, Department of Quality Assurance at LG Institute of Pharmacy, Ahmedabad. Welcome to the MOOC series, initiative taken by LG Institute of Pharmacy. Today we will discuss about the quality control test of the packaging material. In that, we will see about the QC test of containers, closures as well as secondary packaging materials. There are several quality control tests for the primary as well as secondary packaging material. So let's begin with the quality control test for the container. There are different kind of containers like glass container, plastic container, metal container, rubber container. These are the various kind of containers. In the pharmaceutical industry, most of the packaging materials are composed of glass. So, quality control test for the glass containers include chemical resistance test, in that powdered glass test and water attack test, hydrolytic resistance test, arsenic test, thermal shock test, and internal bursting pressure test. These are the 5 types of quality control test for the specific glass container. In the powdered glass test, it is done to estimate the amount of alkali from the powdered glass which happen at elevated temperature. When glass is powdered, leaching of the alkali is enhanced, which can be titrated with using 0.2 normal sulfuric acid, methyl red as an indicator. So we can identify or we can know the amount of alkali leach out from the glass container. In water attack test, it is for type 2 glass. The principle involved in this whether the alkali leaches from the surface of the container or not. In that test, we have to rinse thoroughly container with high purity of water and fill it by 90% of its capacity with water. Autoclave for 121 degrees Celsius for 30 minutes. Then it is cooled and decanted. Liquid is decanted. Okay. And the decanted liquid is titrated with 0.2 normal of sulfuric acid using methyl red as an indicator. And the volume of sulfuric acid is consumed. Is recorded and compared with the limit. So when the result should be within the limit. Okay. If the results are elevated from the limit, it means the container is failed. In the leakage test, drug-filled container is placed in the container filled with the colored solution. Generally, methylene blue solution is used, and autoclave at 121 degrees Celsius for 10 minutes under the pressure. So later on, the containers are observed whether colored get entered into the container or not. If the color appears inside the container, that means there is a leakage and it fails the test. Hydrolytic resistance test only for unused glass container. In that test, we have to rinse the container with CO2-free water for 3 times. Fill till particular volume, okay, and put into the autoclave at 100 degrees Celsius for 10 minutes. Allow to raise temperature to 121 degrees Celsius and then after lower down the temperature at 100 degrees Celsius. Cool it. Specific amount of liquid solution is titrated with 0.01 normal HCl using methyl red as an indicator and perform the blank titration with water and distinguish the difference between the titrations represents volume of HCl consumed by the test liquid, and show the results and compared with its limit. If the result is between the limits, then it passes test, and if the above or below the limits, it fails the test. Arsenic test. We all know about arsenic. It's a heavy metal, right? So it is very important. Okay. In that test, we have to wash inner and outer surface of the container with distilled water, and test solution is same that of the hydrolytic resistance test. Pipette out 10 ml of that solution and add 10 ml of nitric acid. The mixture is maintained on the water bath. Dry the residue in oven at 130 degrees Celsius for 30 minutes and cool it. Add hydrogen molybdate and reflux for 25 minutes. Cool the solution and determine the absorbance at 8, 840 nanometer and perform the blend. So, compare the absorbance with the standard limit. If it complies with the limit, means it passes the test. If it not comply with the limit, it fails the test. Okay. The absorbance of the test solution should not exceed the absorbance of arsenic standard solution of 10 ppm. Okay. Internal bursting pressure test. Name itself suggests pressure test. Means a particular instrument used American glass research increment pressure test. In that test, the test bottle is filled with water and placed inside the test chamber. The internal pressure automatically rises by series of increment at a set time, and bottle are checked at pre-selected pressure level until the container finally bursts. Thermal shock test. In the thermal shock test, sample placed inside the container in upright position in the tray and immerse tray in the hot water for given time. Transfer the container in cold water bath. Temperature should be controlled and examine the cracks before and after the test. Almost 45-degree temperature difference should be there, and the amount of thermal shock a bottle can withstand is based on the construction. Small bottles generally 60 to 80 degrees Celsius and pint bottles 30 to 40 degrees Celsius. So by using this thermal shock test, we can examine how much temperature tolerate the container. There are several tests for plastic container also. Leakage test, same like glass container. Okay, leakage test, collapsibility, transparency, water vapor permeability, and bacteriological test. These are the quality control tests for the plastic containers. Next segment, that is quality control test for the closure. Various tests like sterility test, fragmentation test, self-sealability, pH of the aqueous extract, light absorption test, reducing substances, residue on evaporation, and penetrability. These are the various tests for the particular closure. And the last segment, that is quality control test for the secondary packaging materials. Various secondary packaging materials like cardboard, paper, cartons, corrugated fiber boards. These are the various types of secondary packaging materials. In the quality control test, secondary packaging material designate the packaging used to group various pre-packaged products together. As a secondary packaging is not in direct contact with the actual product, its use and application usually differ distinctly from those of primary packaging. Although the purpose of both types at the same time converge, secondary packaging is said to have two central functions, one that is branding and display and another one that is logistics. In the case of branding and display, secondary packaging play a vital role in the marketing strategy surrounding the product. This is especially relevant in the case of display packaging. In the case of logistics, secondary packaging serves to group several products together for ease of handling, transport, and storage. This means that secondary packaging must be able to contain relatively large volume of primary packaged products, transfer the product safely to the retailer or the consumer destination, and keep the primary packaging in its original condition during the storage. So it's all about the quality control test for container, closure, and secondary packaging materials. Thank you for watching this video. For more updates, like, share, and subscribe our YouTube channel Pharma Ignite. Press bell icon. Thank you.